Hello everyone, let's study Module 7-1. I will talk about cannabis breeding. Genetics is the theoretical foundation of breeding. So I'd like to explain the basic background of genetics through a hypothetical example. Let's imagine there are two trees. One produces red fruit and the other produces green fruit. When these two different types of trees are crossed, the resulting new variety produces red fruit only. In this case, the two parent plants are called T generation, and the new offspring from them is called F1 generation. F refers to the filial generations used in genetics and plant breeding. The F stands for filial, which is offspring in Latin. F1 means first offspring. F2 means offspring of F1. The siblings that F1 has are called dominant allele. The symbol R in uppercase represent red fruit color. However, the symbol for green fruit color, the recessive allele, is written as R in lowercase. And the genetic traits that not visible in F1 but appear in F2 is recessive allele. There are always two versions of every gene. For example, phenotype means the observable traits or characteristics. Homozygous means having two identical alleles. Heterozygous means having two different alleles. Dominant allele is expressed even if only one copy is present, but recessive allele is expressed only when two copies are present. Homozygous means that an organism has two identical alleles for a particular gene. For instance, if a plant is homozygous for the gene controlling seed color, it might have two alleles for green seeds or two alleles for yellow seeds. In both cases, the plant is homozygous because both alleles are the same. Heterozygous means that an organism has two different alleles for a particular gene. Selecting parent plants is the first step of breeding because it determines what traits the offspring can possibly have. We can select two varieties to cross together that are dissimilar. For example, sativa male and indica female. Collect the cannabis plant from the best individual specimens of the finest strains. This slide shows several dominant traits in cannabis. THCA producing allele is considered dominant over the CBDA producing type. Green leaf is a dominant allele over yellow leaves. Palmate leaf is a dominant allele over fused leaves such as duck's foot. Alternate or opposite phyllotaxy is a dominant allele over hold phyllotaxy. Tall height and long internode are dominant alleles over short height and short internode. respectively. These dominant traits are derived from sativa strain. Traditionally, two leaves are produced at each internode. With whole phyllotaxy plants, there are three leaves instead. This can produce a slightly bushier structure to the plants. Whole phyllotaxy is a somewhat fragile genetic anomaly. It usually disappears quickly when attempts are made to stabilize and replicate the property. Just the same as wet cannabis, genetics causes the cannabis leaf to web or mesh together. The result is a leaf which has the same shape as a duck's footprint, hence the name duck's foot cannabis. This unusual leaf appearance is loved by garden growers and outdoor growers, since the duck's foot leaf shape rarely attracts a second glance and easily blends in with natural vegetation. There are many desirable traits in cannabis breeding. For example, for the low THC strain, Santica 70 was developed in France. Top 44 is the quickest flowering strain, is growing just for 44 days or less. Hence the name Top 44. US-31 developed in Ukraine is strongly resistant against leaf spot disease. Cinderella 99, the name was chosen because the plant's exceptional quality, much like Cinderella's transformation. 99 refers to the year 1999, when the cultivar became widely recognized. Cindy 99 is well adapted in Scroll G training. F1 hybrids are vigorous because heterozygous gene combinations perform better and bad alleles are masked. Let's look at the example of a typical F1 hybrid of cannabis, blueberry skunk, in the following slide. Skunk number one is a legendary hybrid cannabis strain that is considered the genetic backbone for many modern varieties. This indica-dominant auto-flowering variety brings the highly prized genetics of a blueberry into the domain of easy to grow and fast flowering auto-flowering plants. Such a feat was achieved by breeding blueberry with the ruderalis variety. Next, let's learn about the breeding process. There is 5 steps to this process. Selecting parent plant is the first step of breeding because it determines what traits the offspring can possibly have. We can select 2 varieties to cross together that are similar, for example, sativa male and indica female. Collect the cannabis plant from the best individual specimens of the finest strain. Selection of parent plants is the first step of breeding of cannabis. Choosing female parents is an easy process than selecting male plants because male plants do not produce resinous flowers, so these traits cannot be directly evaluated in males. Cannabis is a dioecious species, meaning that it has both male and female plants. Pollen is produced by male flowers. These are found on male plants or on hermaphrodite cannabis plants. The pollen tends to be cream and yellow colored and will spread easily in the lightest breeze. Collecting cannabis pollen involves harvesting male pollen sacs. When pollen sacs split often and drying them in a cool, low humidity spot on parchment paper for 1 to 2 days, then gently shaking or taping the branches to release the yellow dust, sifting out debris with a fine mesh screen, and collecting the fine powder for immediate use or storage in a sealed, cool, dry container. Timing is crucial, and preventing contamination by keeping it dry and separate from females is key. Many breeders will use a paintbrush to apply pollen. This can be done with great precision by those who have mastered the art. Backcrossing and selfing are plant and animal breeding techniques to transfer desirable traits and increase genetic uniformity, respectively. Often used together, backcrossing repeatedly crosses a hybrid with one parent to add a new trait, while keeping a good background. While self-filling mates an individual with itself, rapidly increasing homozygosity and creating pure lines, but risking inbreeding depression. S1 stands for first self-generation. An S1 seed is a product of self-pollination, where a female cannabis plant is induced to produce pollen and then pollinate itself or its clone. Backcrossing is to breed a hybrid back with one of its parents, aiming to increase desirable traits from the parent while retaining a specific gene from the hybrid. Each backcross generation is more like parent A, but keeps the desired trait from parent B. The more backcrossing is performed, the higher the genetic similarity to the parents becomes, as shown below. In the BC1 generation, there is a 75% homology. In BC2, there is 88%. In BC3, 94%. And in BC4, there is 97% genetic similarity to the parents. Selfing is a breeding method in which a plant is fertilized using its own plant, producing offspring derived from a single parent. Cannabis is naturally dioecious, so selfing requires artificial induction of pollen. There are some characteristics of S1 Cs. For example, generally they are uniform compared to F1, and they help stabilize traits and often used to make feminized seeds. They are useful in breeding and phenotype selection. They have fixing traits like CBD and THC ratio, flower time. Up to 90% of cannabis seeds lose their viability within a year under improper storage conditions. Therefore, to properly preserve the seeds, they must be stored in a location with optimal conditions. Temperature plays a significant role in seed storage. Seeds should be stored in a cool and consistent temperature between 40 to 50 degrees Fahrenheit to prevent premature aging or germination. Excessive moisture can lead to mold and seed rot, while low humidity can cause seeds to become brittle and lose viability. Exposure to light can trigger growth, so it's essential to store seeds in a dark place. All right, everyone, let's stop here today. Thank you.